So most of you are probably not that familiar with the placenta, so I'll just give a very brief introduction. The placenta is a transient organ, and it's only part of us during intrauterine life, but I'd like to say that really none of us would be here without it, and it really plays a pivotal role for fetal growth and development in utero. So the placenta is on the one side attached to mom. It's attached to the uterus, and on the other side it's attached through the umbilical cord to the baby. And it really serves multiple functions during pregnancy, but probably its most important function is to provide oxygen and nutrients to the growing fetus. So here, I'd like to just say a brief word about what I do clinically. People ask me when they hear that I work on the placenta if I'm an obstetrician, and I'm not. I'm a perinatal pathologist, and you may not have heard of that, but basically what that means is that one of the things that I do is when the obstetrician sees a complication with pregnancy, they send me the placenta to examine after delivery. And I examine this placenta both grossly and microscopically, and based on this examination can tell them most often the cause of the complication, be it bleeding, infection, or something as bad as stillbirth. So one of the common reasons that an obstetrician would send us a placenta to examine is preeclampsia. So preeclampsia is a maternal syndrome that develops during the second half of pregnancy. And it's sometimes called the silent killer because a lot of its symptoms are in fact silent. The two most common symptoms of preeclampsia are high blood pressure, which denotes vascular problems with mom, and protein in the urine, which means that there is kidney dysfunction. Now, there's several variants to preeclampsia. One is gestational hypertension. That's when the high blood pressure exists by itself. There's eclampsia, which means that on top of the high blood pressure and proteinuria, mom develops seizures. There's HELP syndrome, which is, again, on top of these syndromes when mom develops liver dysfunction or low platelets, which means that then she can't clot and she will bleed very easily. So all of these constellation of disorders, they complicate 5 to 8% of all pregnancies worldwide. And as you can imagine, they cause multiple maternal and neonatal complications. So in fact, preeclampsia is the leading cause of maternal mortality in the developed world. It's also the leading cause of fetal growth restriction, or IUGR, which is a bad complication in the fetus. And there's no cure for any of these diseases except to deliver the baby. So in fact, preeclampsia is the #1 cause of induced preterm delivery in the US. So as you might know, babies that are born preterm, and particularly those that are born as growth-restricted babies, they have significant complications, and they will end up having to spend a good amount of their neonatal life, sometimes months, in the neonatal intensive care unit. And this costs approximately $3,500 per day, but it has a bigger cost medically because these babies actually develop so many complications. They can have intestinal problems because they have to be fed through tubes. They can have bleeding in their brain. They can develop what's called retinopathy of prematurity, which can cause them to go blind. And in fact, there are multiple complications they go through, and even if when they survive their neonatal period, when they're adults, they have an increased risk of developing metabolic syndrome and cardiovascular disease. So it's a complication that basically keeps on leading to further complications in adult life. And all of this is because they just had a bad placenta. So what do we know about placental development? Actually, in fact, we don't know that much, especially in humans, about how the placenta develops, particularly early on. We do know, so if you look at this picture of an embryo here, you can see that basically the fetus develops from these group of cells that are on the inside of the embryo. And the placenta develops from this outer layer of cells. And these cells are called trophoblasts. So trophoblasts come from the Greek word meaning to feed. And in fact, that's what the job of these cells is. These cells set out to first establish blood flow from mom to the placenta and then to provide oxygen and nutrients to the baby. So they start out as stem cells in the placenta, trophoblast stem cells, and they have to differentiate further into functional types of trophoblast. And one of these is the invasive trophoblast. This is the cell type that is able to invade the uterus and access maternal blood. And another one is the syncytiotrophoblast, and this is the cell type that is involved with oxygen and nutrient exchange. So let's talk a little bit more about these two types of trophoblasts. So the invasive trophoblasts develop early in pregnancy, and they're perhaps the most crucial to pregnancy success. So if these cells don't develop properly, miscarriage happens, stillbirth happens, and complications like preeclampsia happen. So before pregnancy, mom has these small vessels in the uterus that really can't carry very much blood. So in fact, what these invasive trophoblasts do, they invade the uterus, they remodel these maternal vessels, and they allow for these vessels to carry lots and lots of blood to the growing placenta and therefore to the fetus. The syncytiotrophoblasts, they develop in these functional units in the placenta called chorionic villi. And these units have fetal blood vessels in the center, and they're lined by two layers of trophoblast cells. The inner layer is the stem cell, and the outer layer is the syncytiotrophoblast. So these stem cells fuse to form these syncytiotrophoblasts, which then secrete the pregnancy hormone HCG, and because they're in direct contact with maternal blood, they are able to act to exchange oxygen and nutrients. So when I receive a pre-eclamptic placenta from an obstetrician, I usually find three problems with these placentas. Right away, the placental disc is small, so very much like the babies are small, the placental disc actually weighs very little. It weighs less than 10th percentile for what it should at that gestational age. Then when we cut the placenta and examine the maternal surface, what we find is that there is actually very few invasive trophoblasts. So instead, these maternal vessels are thickened and they're damaged. And then when we examine the chorionic villi, we actually find that these villi have prematurely aged. That means that they have lost their stem cells, and they actually have a high number of these syncytiotrophoblasts, which are shown in brown over here. So in fact, from a trophoblast differentiation perspective, preeclampsia is a disease that has poor formation or function of the invasive trophoblast. And it shows accelerated differentiation into syncytiotrophoblasts. So it's a disease where these trophoblast stem cells prematurely age. And in fact, these syncytiotrophoblasts, because of lack of oxygen, they prematurely age and are not able to do their function. So how can we model this disease in order to be able to find a cure for it or find a way to prevent it? So researchers have tried mouse models, but in fact this disease is specific to higher primates. We can also study the placenta at delivery, but at that point we're only seeing the end stage of disease. And in fact, when we look at that delivered placenta, we can't study the invasive trophoblast because most of those cells are left behind in the uterus. So this begs the question, can we use pluripotent stem cells to model placental development? Can these cells differentiate into these placental cells or trophoblasts? So when we started working with embryonic stem cells over 5 years ago, there was a huge controversy in this field as to whether these pluripotent stem cells in fact can become trophoblasts. Since then, and thanks very much to CIRM funding, we've been able to not only confirm that these cells do differentiate into trophoblasts, but that in fact this process happens through a stepwise process. So in fact, we can first get trophoblast stem cells differentiated in the dish, and subsequently these cells will differentiate further into the invasive trophoblast and the syncytiotrophoblast. And we've also been able to find a mechanism for how this process occurs in culture. And finally, we've been able to find a defined culture condition for this stepwise differentiation. So the next question after this, though, is that can we in fact model a trophoblast defect in culture? So for this, we actually turned to trisomy 21. Trisomy 21, most of you might know as Down syndrome. And you might also know that Down syndrome patients have complications with their heart. They have neurologic abnormalities, but what you may not know is that babies that are born with trisomy 21, they also have defects with their placenta. And in fact, the higher rate of miscarriage with trisomy 21 pregnancies is due to these placental abnormalities. In fact, what researchers have found is that the trisomy 21 placentas have a specific defect where they can't form syncytial trophoblast. So we asked, what if we take trisomy 21 iPS cells and differentiate them and ask, are they able to differentiate into syncytial trophoblast? And in fact, we found that the fusion index of these trisomy 21 iPS cells is much less when you compare to a normal karyotype iPS cell. And these trisomy 21 iPS cells also retain their trophoblast stem cell markers longer, and they show a delay in secretion of the pregnancy hormone HCG. So all of this basically provided proof of concept to us that we can, in fact, model a trophoblast defect in culture using iPS cells. So now going forward, and again, thanks to CIRM funding, we now have a project underway to model the invasive trophoblast defect of preeclampsia using iPS cells. We are recruiting patients in the labor and delivery unit at UCSD, consenting these patients for sampling their placentas. And from these placentas, we isolate amnion cells. This is a layer of cells that is on the fetal surface of the placenta. And what we plan to do, we're banking these cells, and we plan to generate iPS cells from these amnion cells. And once we have those iPS cells, we can ask what is their potential to differentiate into these trophoblast subtypes. And we hypothesize that they are going to show a defect in either differentiation or function of these invasive trophoblasts. And once we have that, we can actually use these cells in order to screen for drugs that promote differentiation into this lineage and hopefully be able to either prevent or treat preeclampsia. So in order to do this, we have put together a strong interdisciplinary team of clinicians and scientists, and these include Dr. Louise Laurent, who is a high-risk obstetrician and a stem cell researcher at UCSD. We work very closely together to consent these patients and to generate iPS cells. And also Dr. Robert Oshima, who is from Sanford Burnham Institute for Medical Research, and he is an expert in the area of drug screening and high-throughput assays. So we also have a very large group of researchers that are working on this, and I'm very thankful to all of them, and also very thankful for our funding, particularly to CIRM, who has really provided support for us for this project from the very beginning.